Hi, welcome to a laboratory session on Ouchterlony double diffusion pattern and Ouchterlony double diffusion titration. And because I just melted the agarose, I will begin the experiment by showing you how to pour the agarose on a slide, and then while it is solidifying, I'll continue the experiment. I'll be bilingual for the benefit of the local students. Hopefully, I can do this. So that's how we pour the agarose, molten agarose on a slide. And as long as the center is neatly covered, we do not disturb it. And just because the agarose is there, still in its molten state, I'll show you how to pour it on another slide as well, so that you get the hang of the things. And then I'll go ahead with demonstrating or speaking about the experiment. It's a little messy, but yeah, I did not want to waste the agarose. So ఇవాళ నేను మీకు Ouchterlony ప్యాటర్న్ అండ్ Ouchterlony డబుల్ డిఫ్యూజన్ అనే ఎక్స్పెరిమెంట్స్ చెప్తాను. అందులో ఫస్ట్ మనం చేయబోయేది గ్లాస్ స్లైడ్ మీద అగరోజ్, మెల్ట్ చేసిన అగరోజ్ ని పోర్ చేసి ఎలా సాలిడిఫై చేస్తాము అనేది చూసాక వి విల్ కంటిన్యూ ది ఎక్స్పెరిమెంట్. ఇది మాల్టన్ అగరోజ్. ఎలా ప్రిపేర్ చేయాలి, ఏం చేయాలి అనేవన్నీ తర్వాత చెప్తాను. ఇలా మనం పోర్ చేసి స్లైడ్స్ అయితే రెడీ చేసుకున్నాం. Yeah, now because I'm done with the most important part of the experiment, now we'll coolly go into the explanation part. Yeah, Ouchterlony was the one who came up with this technique for identifying the similarities between different antigens from different sources to a particular antibody. And because Ouchterlony is the one who has come up with this technique, it's called Ouchterlony diffusion, and it's a double diffusion technique. So it's called Ouchterlony double diffusion. And I'll explain that in a minute. So we'll begin with the experiment. The point of doing Ouchterlony pattern, first we'll do the pattern, and for doing the pattern, this is a slide on which I already poured the agarose, and the agarose is solidified. What we'll be needing for performing Ouchterlony double diffusion Ouchterlony pattern is a slide on which we have poured a layer of agarose and which has been solidified. It should not be dry, it should just be solidified. So the slides should be prepared fresh before each experiment. And then we will be needing the 1x assay buffer, and then we will be needing agarose, which is also provided in the kit. We will dilute this 10x assay buffer to 1x before we prepare the agarose solution. This is a 10 ml medicine cap. As you can see, a syrup, it comes with a tonic, and I have the agarose has solidified, so it must be solidified on the plates as well. But before we move them, we will finish the explanation. For performing Ouchterlony double diffusion pattern, we will the kit is provided, and the kit has antigen X1, antiserum X, and antigen X2 also. Antigen X2. So what we do is, first we pour agarose on a slide. Once the agarose solidifies, the gels have to be punched with a gel puncher, and the pattern is already given. That's called the template. So before you begin the experiment, you'll be needing the template and the gel puncher. I'll also show you how to punch the gels on an already solidified gel. So we have the template. You put the template on your working desk and gently put your slide on the template so that you can see it clearly. The whole thing looks like this. Once the agarose is, because the agarose is transparent, we can see the, and then carefully on the circles that have been printed on the template, you put your gel puncher all the way to the bottom, pull your syringe out and gently remove it. Here we have a hole and we can test whether the hole has come properly or not by inserting a tip. Again, repeat till you finish punching all the holes. For this slide, I'll punch all the holes. When we are doing titration, it is a similar pattern, so I won't be explaining so much. Just make sure it comes off. So that that is the hole we will fill with our antibodies and antigens. And while I'm punching, I'll also keep explaining. Now in Ouchterlony pattern, what we see is, suppose we have a source which has lots of antigens, or you're expecting a lot of antigens, or you got a new disease and you want to identify what antigen it could resemble with. Suppose we have, for COVID, we had cough, cold, influenza, a type of symptoms. So to identify whether it is similar to influenza or not, probably this can be done. I don't know, that's a guess I'm taking, but I'm sure it's a good guess because that's how we apply it. You try to identify antigens which have similar properties. So we have finished punching the gel. You can see it clearly. This is for pattern identification. Now, what is the pattern that we are going to identify? We are going to identify if two antigens share antigenic epitopes or do not share antigenic epitopes or partially share antigenic epitopes. Simply put, we will identify whether the antigens are similar or not. Now, why should you identify if antigens are similar or not? Sometimes we'll get I can't say new diseases, different sorts of diseases, but with similar overlapping symptoms and all. So to start off treatment and all, it's better to know if they are similar to the previous antigens already known and already established, so that it's easier to go ahead and establish tools of identification. And also, if we have a large pool of antigens, it's better to know their identity or similarity. ఈ Ouchterlony డబుల్ డిఫ్యూజన్ ప్యాటర్న్ అనే ఎక్స్పెరిమెంట్ ఈ విధమైన స్లైడ్ మీద చేస్తాము. జెల్ పోర్ చేయడం నేను మీకు ఆల్రెడీ చూపించాను, పంచ్ చేయడం కూడా చూపించాను. పంచ్ చేసాక ఆ స్లైడ్ ఇలా కనిపిస్తుంది మనకి. ఈ స్లైడ్ వాడి మనం ప్యాటర్న్ ఐడెంటిఫికేషన్ చేస్తాం. దేని ప్యాటర్న్స్ ఐడెంటిఫై చేస్తాము? మనము రెండు యాంటీజెన్స్ కి సిమిలర్ ప్రాపర్టీస్ ఉన్నాయా, అవి రెండు సంబంధం లేనివా, కొంచెం దగ్గర సంబంధం ఉన్నవా, లేకపోతే కొంచెం మ్యాచ్ అయ్యి కొంచెం మ్యాచ్ అవ్వకుండా అలా ఉంటాయా అనేది ఐడెంటిఫై చేస్తాం. అది ఐడెంటిఫై చేయడం ఎందుకు ఇంపార్టెంట్ అంటే, మనకి కొత్త కొత్త యాంటీజెన్స్ వచ్చినప్పుడు, మన దృష్టికి వచ్చినప్పుడు, వాటిని ఇదివరకు ఉన్న యాంటీజెన్స్ కి ఏమైనా సంబంధం ఉందా లేదా అని చెక్ చేయడానికి ఇది ఉపయోగపడుతుంది. Once we do this experiment, in the end result will look something similar to this. Okay, we will either get a pattern which is like a X or a Y or a V sort of a pattern. So what do these patterns indicate? X indicates no identity. The antigens do not show similar antigenic epitopes. The antigenic epitopes on the two different antigens are not related, they are unrelated antigens. Then we have Y. This Y, what Y indicates is they're partially similar. కొంచెం దగ్గర సంబంధం ఉన్నావు కానీ కొంచెమే సంబంధం ఉన్నావు. ఇది ఎక్స్ ప్యాటర్న్ చూపిస్తే అసలు ఐడెంటిఫైబుల్ కాదు, ఆ రెండు సేమ్ కాదు. వై ప్యాటర్న్ చూపిస్తే అవి రెండు కొంచెం సేమ్, కొంచెం దగ్గర ఉంటాయి కానీ మరి అంత దగ్గరవి కాదు. ఇలాంటి ప్యాటర్న్ వస్తే అవి రెండు సేమ్, చాలా సిమిలర్ అన్నట్టు, చాలా సిమిలర్ గా ఉన్నవే ఇలాంటి ప్యాటర్న్ చూపిస్తాయి. So we will have three patterns, X, Y and V, as you can see here. And X says pattern of non-identity, Y is partial identity, and V is the other one is good identity. So how we go ahead with the experiment is, we will take the Ouchterlony double diffusion pattern kit. I have used the Ouchterlony double diffusion pattern kit, and in the kit we are supplied with all the antigens, two different types of antigen for each antibody. We are trying for three different antibodies, and the antigens will be loaded in the bottom with two antigens here, two antigens here, and two antigens here: antiserum, antiserum, and antiserum. So we will load 20 microliters of antiserum. But before you load the antiserum, you have to, after loading the antiserum, the whole point of doing this experiment on an agarose gel is you allow the movement of the antibody and the antigen. They keep moving through the pores in the agarose gel. Agarose gel enduku vaadtamu ante ee experiment ki antibody antigen kuda ఒకదాని వైపు ఒకటి మూవ్ అవ్వడానికి ఈ పోర్స్ లోంచి వెళ్లే ఆస్కారం ఉంటుంది. సో అవి ఒకదాని నుంచి ఒకటి డిఫ్యూజ్ అవుతూ ఒకదాని దగ్గరికి ఒకటి వచ్చినప్పుడు అవి విజిబుల్ రియాక్షన్ ఫామ్ చేసి ఒక గీత లాగా కనిపిస్తాయి. దట్స్ హౌ వి వర్ ఏబుల్ టు సీ ఇట్, విజువలైజ్ ఇట్ లైక్ దిస్. ఒక గీతలాగా కనిపిస్తాయి. దాన్ని ప్రెసిపిటేషన్ అంటాం. ఏబిఓ బ్లడ్ గ్రూపింగ్ లో ఎగ్లూటినేషన్ రియాక్షన్ ఈ పర్టిక్యులర్ ఆక్టోలోని డబుల్ డిఫ్యూజన్ ప్యాటర్న్ లోని, డబుల్ డిఫ్యూజన్ టైట్రేషన్ లోని కూడా మనం వాడే ప్రిన్సిపల్ ని ప్రెసిపిటేషన్ రియాక్షన్ అంటాం. యాంటీజెన్ యాంటీబాడీ సమానమైన కాన్సన్ట్రేషన్స్ లోకి రీచ్ అయినప్పుడు ఒక విజిబుల్ లైన్ ఫామ్ చేస్తాయి. ఆ విజిబుల్ లైనే ప్రెసిపిటిన్ లైన్. The whole principle behind this pattern is through the agarose gel, the antibody and the antigen diffuse, and when they come to a point of equivalence, they form the precipitin line, which is visible. So the reaction takes time to happen, so we incubate it overnight at 37 degrees in a moist chamber. Now, moist chambers, because we do not have a ready-made moist chamber, I have just taken a small box and I have used cotton. So what you do is you spread the cotton well in the box like this, wet it a little so that it is moist, and you have your moist chambers ready. So you can wet it like this with cotton, and this is the bottom part of your moist chamber. Similarly, we do it with the lid. so that it is sealed properly on both ends, and we have a moist chamber in which the reaction can take place. Why should it be moist? ఎందుకు మనం దీన్ని తడిపి పెట్టాలి, ఎందుకు ఇది వెట్గా ఉండాలి? Because only if it is moist the agarose will be not dried up, and the agarose will allow the movement of the antigen and antibody within it. Moist ga lekapothe ఈ అగరోస్ డ్రై అప్ అయిపోతుంది. అగరోస్ బాగా డ్రై అప్ అయిపోతే ఇంకా మూవ్ అవ్వలేదు, మన యాంటీజెన్ యాంటీబాడీ మూవ్ అవ్వలేవు ఒకదాని నుంచి ఒక దాని వైపు. అందుకని మనము ఇది కంపల్సరీ ఈ చేంబర్ మాయిస్ట్ గానే ఉంచాలి. సో ఫస్ట్ వీ విల్ ప్రిపేర్ ద మాయిస్ట్ చేంబర్ లైక్ దిస్, అండ్ దెన్ వీ కెన్ పుట్ ఇట్ ఆన్ లైక్ దిస్. అండ్ వన్స్ ద మాయిస్ట్ చేంబర్ ఇస్ రెడీ, యూ కెన్ start loading the antibodies and antigens. They are listed as anti-serum antigen X1, X2, and then you also have the antibody X, anti-serum X, anti-gen X1, antigen X2. First you pick that, and then I'll just show you one set, and then I'll load the others. I hope you can see this. It is anti-serum anti-serum Y, and then you have antigen Y1, and then you have antigen Y2. So what we'll do is we will start loading. The antigens go in the bottom wells, and the anti-serum goes in the top well. And once they are provided, so we finished loading this, so we'll put this the other side. Then we have antigen Y1. Then we have antigen Y2. Gently load it into the well. Similarly, you load all the other wells also with X1, X2, and X3, and Z1, Z2, and anti-serum X, anti-serum X1, X2, and X3. This is anti-serum, so it goes in the top well. Against this anti-serum, which is produced against a known antigen, we are testing antigens of our choice to see if our test antigens show any similarity with each other against the anti-serum used. Okay. So that's how we load all the wells. And once the loading of all the wells is done, you can just gently put it in your moist chamber and leave it at room temperature overnight without disturbing. You can put it at a place where you won't disturb it overnight, so that the reaction, the diffusion of the antibody and the antigens takes place through the agarose properly till a visible Presbyterian line is formed. And the main thing here is the pattern of identification. X is here how we say is, this is pattern of identity, this is pattern of partial identity, this is pattern of non-identity. So once you get patterns like that, you can say whether your antigens of choice or antigens which you have picked show any antigenic epitope identity with each other or not. మనం జాగ్రత్తగా ఎక్స్పెరిమెంట్ చేసేసుకున్నాక దాన్ని అలాగా మాయిస్ట్ చేంబర్ లో పెట్టేయాలి. పెట్టేసి రాత్రంతా రూమ్ టెంపరేచర్ లో వదిలేయాలి. వదిలేసాక మనకి ప్యాటర్న్స్ కనిపిస్తాయి. ప్యాటర్న్స్ కనిపించినప్పుడు మనం ఆ ప్యాటర్న్స్ జాగ్రత్తగా ఇంటర్ప్రెట్ చేసుకోవాలి. ఇక్కడ మన పర్పస్ కోసం, ఫర్ టుడేస్ ఎక్స్పెరిమెంటల్ పర్పస్, నేను ఇది వి అంటున్నాను, ఇది ఐడెంటిటీ. ఇలాంటి ప్యాటర్న్ కనిపిస్తే ఆ రెండు యాంటీజెన్స్, మనం దేన్నైతే టెస్ట్ చేస్తున్నామో ఆ రెండు యాంటీజెన్స్ ఒకదానికొకటి రిలేటెడ్ ప్రాపర్టీస్ ఉన్నవని. ఇలా వస్తే పార్షియల్ ఐడెంటిటీ, దే ఆర్ ఓన్లీ పార్షియలీ సిమిలర్ టు ఈచ్ అదర్. ఇలా వస్తే అవి రెండు నాట్ ఐడెంటికల్ టు ఈచ్ అదర్, ఆ రెండిటికి యాంటీజెనిక్ ఎపిటోప్స్ కి సంబంధం లేదని దాని అర్థం. దిస్ ఇస్ ద పాయింట్ ఆఫ్ దిస్ ఇస్ హౌ ద Ouchterlony pattern looks, and it's based on precipitation. Now, see whether it's Ouchterlony pattern or Ouchterlony titration, the gel preparation is the same. The only difference is the templates. So I have finished punching this gel also. I'll quickly explain and then we will complete. Now, if Ouchterlony pattern is for identifying the antigenic epitope similarity, Ouchterlony titration is for identifying the concentration of the antibody that can react with the test antigen. We have a test antigen which we load in the center well, supposedly center well, and then we will put different concentrations of antiserum all around, and then we will see at what minimum concentration the antigen can still show reaction with the antibody. So it's also very similar. We will load the anti-serum in the here the point is serial dilution. These gaps are taking up a lot of time, but I'll just show you. I hope you can see the way I numbered these tubes. So see, these tubes are numbered this way, so that when I make serial dilutions of the antibody, we will know at what minimum concentration the antigen-antibody reaction can still take place. If you can notice, I have labeled them and I'm holding them in my hand like this in this pattern. So here's the antibody to begin with. In the first one, it will be undiluted, so I directly put it from that. If you can see, I put 1x assay buffer in all this. Now, in case I missed telling you how we prepare 1x assay buffer, the 10x assay buffer will be given in the kit. Then what you do is you take a measuring cylinder, pour 1 ml of this assay buffer into the measuring cylinder and make it up to 10 ml with distilled water. That is 1 ml of assay buffer, 10x assay buffer plus 9 ml of distilled water will give you your 1x assay buffer 10 ml. In that, now to also tell you how to prepare the agarose solution. Now the agarose solution also is prepared in 1 ml, 1x assay buffer, 10 ml of 1x assay buffer based on your plate capacity. I took 10 ml of assay buffer. Into that 10 ml of assay buffer, you put zero 0.1 gram of agarose, and then you melt it till the agarose is completely molten, and then you cool it down to room temperature, not room temperature, cool it down to 50 degrees or so, and then pour it on the plates and wait for it to solidify. That's how we prepare agarose solution for pouring on the plates. And in these, for dilution in Ouchterlony titration, I took 1x assay buffer, 20 microliters in each tube. And now I am serial diluting. From this, I take and I put it in this, mix it, discard the tip, rest it. From the second tube to the third tube now. I hope this is visible. Then mix it. So we are ready with a serial diluted anti-serum. And then from the last tube, you just discard the last 20 microliters. Now we are ready. So we'll fill it in all the wells labeled E, F, B, C, D. E, F, B, C, D, one by one. So we are starting with E, and here we can put a mark with the marker somewhere here, a small dot to say which side is D. Maybe I'll write D here. I wrote D on the edge of the slide, as you can see. Not a beautiful plate, but plate all right. Okay, this is the other plate which has agarose on it. So you can use both, or you can use one. So I'm starting with this here. From each tube, I'm loading one concentration after serial dilution. Here also agarose is used because it allows the movement of antigen and antibody towards each other, diffusion, and then when they come to a zone of equivalence, they will. So now that we finished loading, we will discard these tubes, and in the center, we will take the antigen. What we can do is now again put it in a moist chamber. Again, incubate it overnight. Now, after you incubate it overnight for Ouchterlony double diffusion pattern, it will look something like this. Something like this. And then based on the pattern, you can identify the least concentration of antibody that can still react with the antigen. And here, as you can see, there is no Presbyterian line formed between this antibody and the antigen in this interval. So this is a concentration that cannot react at all. So that's how we identify titrations, or we can identify the minimum amount of antibody that can react with the antigen, or the minimum amount of antigen that shows reaction with the least quantity of antibody. That's for your Ouchterlony double diffusion titration. And pattern. Pattern on the slide can be seen like this. This is a pattern which is showing antigens with similar antigenic epitopes. This one. So I hope you understood double diffusion in both titration and pattern as well, and hope to come back soon with another laboratory session. Till then, thank you.